Welcome to the Enzymes and Energy chapter, or Energy and Enzymes, I guess is how it reads in the book. If you're following along in a text, I'm using Campbell 10th edition. This is chapter 8. Any Campbell Biology edition works, but Energy and Enzymes. So that's what we're going to talk about. This image is bioluminescent fungi, and we're going to talk about the energy that produces bioluminescence. It's pretty cool. But I think that's a really neat photo. They look like jellyfish to me a little bit, but they're not. They're bioluminescent fungi, and I thought they were a great opener to this chapter. So, quick fun fact overview. I like these at the start of all of my chapters. Remember that living cell is this little miniature chemical factory, and there are thousands of reactions occurring all the time, where you're just giant little chemistry experiments going on. Every living cell is. That cell's going to get energy and it's going to apply energy to do work. So this is really about the production and consumption, or the acquisition and consumption of energy. Some organisms can even convert energy into light, and that's that bioluminescence we just saw in the last slide in those fungi. Remember to grab your definitions, your bolded word definitions. So metabolism, when we talk about this from a purely biological and not a nutrition standpoint, when we talk about metabolism from a biological standpoint, we're talking about all of the chemical reactions that happen within an organism. So everything endergonic and everything exergonic, all of it. And there's pluses and minuses there, and we're talking about the sum total. Do these balance out at zero? Do I have more that release energy? Do I have more that use energy? What's going on here? Metabolism is an emergent property. So if you've forgotten what an emergent property is, head on back to, I don't know, I think it's chapter 3 or something. Maybe it's chapter 4, I don't remember. But head on back over there and refresh. Remember what emergent property means. But what happens is we get all these interactions between molecules within a cell. So we talk about metabolism, we need to talk about a metabolic pathway, and that's just starting with a specific molecule or a specific reactant and ending with a specific product. Each step along that way in that metabolic pathway is going to have the potential to be catalyzed or sped up by an enzyme. So we have catabolic pathways and anabolic pathways. Remember back to the macromolecules chapter when we talked about catabolic and anabolic reactions. We talked about catabolism and anabolism. I think in my slides I use Captain America because he's my favorite superhero. I use Steve Rogers images from the movies to help you reinforce catabolic and anabolic. So we're going to take it just a little bit further. We're going to give you a little bit more information. So catabolic pathways are going to release energy. They're going to break down complex molecules into simpler ones. We're going to do this in cellular respiration. So we're going to take glucose, we're going to break it down in the presence of oxygen, and we're going to modify it, we're going to change it, and we're going to yield ATP from it. Anabolic pathways are going to be those that consume energy. They're going to use energy to build more complex molecules from simpler ones. So this is going to be that image of Steve Rogers going from tiny Steve Rogers to Captain America Steve Rogers. We're going to synthesize proteins from amino acids. We're going to take those simple molecules, those amino acids, and we're going to build them into bigger ones, into bigger, more complex proteins, and that is anabolism. Bioenergetics, another vocab, bioenergetics is the study of how organisms manage their energy resources. How do they do this? What do they do? Where do they get their energy from? Where does it go to? How do they manage this? So let's talk about energy. Energy is the capacity to cause change, and we can have energy in a bunch of different forms. Here's some. This is not by no means all. These are base definitions, so kinetic energy versus the third one down is potential energy. Kinetic energy is the energy associated with motion. I'm going to skip to the third one for a second. Potential energy is going to be that energy that matter has because of its location or its structure. So, and by its structure, I mean its chemical bonds. What are its bonds? How are its bonds formed? How much energy is in those bonds? How much energy can I get if I break those bonds? Back up to the second bullet. Heat. Heat is thermal energy. So it's a form of kinetic energy. It's thermal energy. And it's the kinetic energy that gets associated with the random movement of atoms or molecules. We talked about atoms and molecules moving randomly when we talked about diffusion. So we're going to get kinetic energy from that process. Fourth bullet is chemical energy. Chemical energy is that potential energy available that is available to release in a chemical reaction. ATP to ADP. Okay, that's chemical energy. I'm going to get that energy when I break that bond. So you've got solid definitions for kinetic and potential energy. You've got examples of each, thermal energy being or heat being an example of kinetic energy, chemical energy being an example of potential energy. And then we have a visual coming up for this in a moment. But energy, remember that we can convert energy from one form to another. So I can take potential energy, I can convert it into chemical energy. We have heat energy, we can convert it. I can have potential energy that gets converted into kinetic energy. Kinetic energy can go through motion and then become potential energy again. And that's what this actually demonstrates for you. So this is showing you that that diver has more potential energy up on the platform. It's going to convert that potential energy into kinetic energy through that motion as it dives into the water. In the water, it has less potential energy than it had on the platform. And then as it climbs those stairs, it's going to convert the kinetic energy of those muscle movements into potential energy when it gets back up to the top of that platform. All right, so we're going to do just a little bit of whiteboard matching here. Let's get some of these definitions knocked out of the way. You're going to pause. I'm going to count to, I don't know, 5, 7, 10, whatever I count to before I realize I'm still counting, and then I'm going to advance the slide. How'd you do? I swear I did not try to make energy match with E for energy or heat to match with H for heat or C to match with C for catabolism. That was purely random, but I think it's kind of funny that that's how it worked out. All right, thermodynamics. Thermodynamics is the study of how energy gets transformed, okay? So for a moment, I want you to think about a not biological situation. I want you to think about a thermos. I want you to think about a closed system, okay? In a thermos, the contents inside that thermos are isolated from their surroundings. That's the whole point of a thermos. It's so that we can retain heat or cold in a space and not let that energy be lost to the environment around it. In an open system, so I don't get an energy transfer there, it's isolated. In an open system, energy gets transferred. It gets transferred between the system and its surroundings. It's the liquid in your coffee cup and not in your thermos. Okay, in your coffee cup, it's going to lose heat faster because it has an interaction with the environment, and that energy transfer goes from the liquid into the atmosphere. That doesn't happen in your closed system thermos. Remember that organisms are open systems. They are not closed systems. Closed systems happen in other places, and in biology, there's always the impact of the surroundings. So even when something's supposed to work the way it does, it's, you've worked it out on paper, this is what should happen. That doesn't always happen because we're talking about open systems. Okay, we need to look at thermodynamics and the first two laws. So the first law of thermodynamics, you've heard a gazillion times. You've heard that energy cannot be created nor destroyed. Okay, and that's true. But the other part that nobody ever says is that it can be transferred and transformed. So energy can be transferred from one molecule to another. Energy can be transformed from one form to another, potential to kinetic and vice versa. So the first law of thermodynamics is also called the law or the principle of conservation of energy. So if you've learned it that way, that's fine. Just know that those two things are interchangeable. The second law deals with when that energy transfer or transformation occurs. Okay, so every time we transfer or transform energy, some of it is unusable. And then put some air quotes here, it's lost as heat. Now it's not really lost because we can't destroy it. It's just not usable to us. Okay, It gets moved into a different form that's not currently usable to us. It's not actually leaving the ecosystem. It's just, or even the biosphere rather, is probably a better way of saying that. It's just being transferred around. So the second law of thermodynamics says that every energy transfer or transformation increases the entropy. Entropy is disorder. I want you to think of it as disorder as like chaos, okay? The disorganization. So every energy transfer or transformation increases the disorder of the universe. So every time we quote unquote lose some energy as heat, we're making the universe or the biosphere less organized. Okay? It just means it's not usable to us. Okay. Entropy comes back later in an equation, and that's why I introduce it to you all here. All right. So this whole order and disorder thing, right? So cells, remember we said one of those 7 structures or one of the 7 characteristics of life was that those cells had ordered structures, right? And we had ordered materials. And they create those things. They create structures from less organized or less ordered materials. We said that they replace ordered forms of matter and energy with less ordered forms. Okay, and we're going to talk about that in a second. And then we talked about energy flow. So here's what I want us to understand. I want us to understand that living cells convert organized forms of energy into heat. This is not an avoidable situation. This is what happens. This is a byproduct of life. There's no stopping it. The only way to stop it is to not be living. Okay? That I want us to understand that spontaneous processes occur without any energy input. They can happen quickly or they can happen slowly. Spontaneous does not mean fast. Spontaneous means that it happens without energy input. Okay, and so for a process to do that, for a process to occur without an energy input, it must increase that entropy in the universe. We must follow the laws of thermodynamics. If I'm not going to put any energy in, I'm absolutely going to lose energy in the form of heat, and that's just the way it is. Evolution, so this is an argument that comes up, I don't know, probably every couple of semesters by somebody who's taken another course and they're really working this through, and they'll say to me, well, that doesn't make any sense. Evolution, so we're evolving into more complex organisms, and that violates the second law of thermodynamics because second law of thermodynamics says that we increase the disorder, and evolution says we're increasing order, and that's just not true. Evolution as a process does not violate the second law of thermodynamics. Nobody's violating any laws, at least not in this classroom. So it doesn't work that way. We're just transferring. And so the entropy or the disorder in a single organism may decrease through that evolutionary process, but the universe's total entropy increases. So, and that's the key to that. So, I just want to make that clear because it pops up now and again, and surely there's somebody sitting out there going, but wait a minute, Dr. P, what about? Yeah, I know that it seems that way, but it's actually not that way. So we need to talk a little bit more about free energy. So free energy, changes in free energy within a reaction tell us whether or not that reaction is spontaneous or not. Remember your definition of spontaneous. Spontaneous is not fast. Spontaneous can be quick or it can be slow. All spontaneous means to us here is that I don't have to put energy in to make the reaction happen. Okay? And you're like, yeah, so what? Who cares? Well, biologists care because if we know which reactions happen spontaneously, we know which reactions require an input of energy and which ones don't. And by doing that, we're able to determine energy changes that occur within chemical reactions. Is this one going to be endergonic or exergonic? Am I going to get energy from this? I'm going to have to put energy into it. And doing that helps us see how it all balances out, how one feeds the other. And that's going to become important here in a moment. So here's your definition for free energy. Free energy is that energy that can do work when temperature and pressure are uniform. So we're talking like within a living cell. So within that cell, that one cell where those chemical reactions are going on, the pressure and the temperature are basically uniform. Okay? It's not varied across the cell. All right. Yeah, just a little math. Don't panic. Okay. Delta, that triangle is a delta. Delta G, and delta means change in. So delta G equals delta H minus T delta S. Okay, so delta G, G is your free energy. So delta G is the change in free energy. Okay, so the change in free energy in a process is related to the change in total energy. So total energy is delta H. Change is delta, H is total energy, minus the temperature, T is temperature in Kelvin, multiplied by delta S. Delta, again, is change, and S is the amount of entropy. So you need to know that equation so that if I give you, all but one of those variables, you can tell me what it is. So if I give you delta G, delta H, delta S, you need to be able to tell me the temperature in Kelvin, or vice versa. Why is this happy little equation important? Right? You're like, okay, great. I got to memorize this. Now I got to do some math. I'm freaking out, but I don't even know why I'm doing it. Here's why you're doing it. When you get a negative answer for delta G, when delta G is negative, when the change in entropy is negative, that reaction, that process will be spontaneous. It means I don't have to put in any energy to make it happen. Okay? That's important because if I have a spontaneous process, I'm going to release energy from it and I can use that energy to perform work. So I'm looking for the spontaneous reactions because those are the ones that are going to drive work for me. I can harness it to perform work. I'm still going to get work product out of a non-spontaneous reaction, but I've got to put energy into it to accomplish it. So I got to pay something to get something. In this case, I don't have to pay anything up front. I have the energy stored in those molecules as potential energy, and I'm just going to get the work basically for free. So graphically, it looks like this, okay? So on the left is an exergonic reaction. It's going to be a spontaneous reaction. So on the left, the vertical axis of this graph is the free energy. And on the bottom, the x-axis is the progression of the reaction. So I'm going to move from reactants to products. And the reactants have more free energy in them. That bar is higher on the graph, right? So there's a lot of potential energy in those reactants. And as those reactants move spontaneously, I don't have to add anything to this. They're spontaneously going to move to their product form. Those products are going to have less free energy. They're lower on the y-axis. See that? They're lower. They're down. I have an amount of energy that's released. Okay? So I move from a bigger number to a lesser number. So if I call the reactants, the level of reactants 0, and I move towards the product, now I'm less than 0, and I've gained energy from it, and I've gotten the product. And this has been great. This is spontaneous. On the right, the blue graph is just the opposite. The energy in my reactants is less than the free energy in my products. And in order to go from those reactants to those products, I have to absorb energy. I have to spend energy to make that happen. This is called an endergonic reaction. Ender, into. I have to put energy into the reaction from its surroundings. And oftentimes that can be heat energy, that can be ATP. I got to put energy in. These guys are considered non-spontaneous. So exergonic reactions proceed with a net release of energy. They're considered spontaneous. Endergonic reactions absorb free energy from their surroundings, and they're considered non-spontaneous. I have to put energy in to make that reaction go. All right, so at this point, you should be able to tell me what does delta G mean and what is free energy. You guys pause, I'll count to 5 and we'll advance. How'd you do? Got another one for you. You ready? If you're playing at home by yourself, do both sets of tables. It's set up this way for my classroom. So those of you in the classroom, you know that. Those of you not in the classroom with me for lecture, you don't necessarily know that, but just play along both sides for yourself. All right, you guys pause. I'm going to count to 10. Those should look familiar. And I believe I got all the labels on there. Products, reactants, energy, free energy, progress of reaction, and delta G. Should all be good. Okay. All right. So, back to the ATP. Love the ATP. So, ATP, remember what we said, it was the energy currency for cells. It's the primary form of energy that cells use to accomplish work or to accomplish something. Okay, and ATP does that by coupling an exergonic reaction to an endergonic one. So one that releases energy to one that needs energy. Okay, so it's like money. I go to the store and I want to buy an Xbox. I have to have X amount of money to do that, right? I have to have a certain amount of money to buy that Xbox. If I don't have enough money, I can't buy that Xbox. If I don't have enough energy, I can't run that endergonic reaction. So the cell has three main kinds of work that it does. It either does chemical energy, it does transport, or it does mechanical energy. You should be able to think of examples of both. If you can't, I want you to hit back into one of your textbooks and find an example of both. I'm not going to tell it to you. I want you to go back. We've talked about all three in the over this course, perhaps not this chapter exactly, but we have talked about it. So I want you to go back and find it. I want you to start building your knowledge together. So to do work, cells have to manage their energy resources. We had that definition, managing energy resources, the study of how cells or organisms manage their energy resources. Go find that definition. But they do that through energy coupling. And what happens is they use an exergonic process to drive an endergonic one. So the definition of energy coupling is using an exergonic process to drive an endergonic one. Most of that energy coupling is done through ATP. Remember your structure of ATP. Okay. All right. When I break the bonds between the phosphate groups on ATP's tail, which I do through hydrolysis, I'm going to release energy. I'm going to release the most bang for my buck, the most energy when I cleave off the third from the second. I'm going to get some energy when I cleave the second off from the first, right? But not as much. I still get energy, just not as much. Okay, and what happens is that energy comes from the chemical change. I'm lowering the state of free energy. It's not the bond itself, it's the amount of energy possessed with those electrons. There's your image for my visual peeps. Sorry about that. That was guinea pigs bumping their dishes together. Okay, this is how it works. So remember, we've got three types of cellular work: mechanical, transport, and chemical. These guys are powered by the hydrolysis of ATP. In the cell, the energy from the exergonic reaction of ATP hydrolysis, so cleaving off a phosphate, releases energy, and then we use that energy to drive some other process that needs an energy input, an endergonic one. Overall, big scheme pictures, I have to have exergonic reactions. Overall, I have to have more exergonic than endergonic. I can't have more endergonic than exergonic because that means I need more than I have, I need more money than I have, and the end product isn't going to be the same. Okay? So how we do this is we drive those endergonic reactions through a process called phosphorylation. Phosphorylation is when I transfer a phosphate group to some other molecule, some other reactant. And whoever received that phosphate is now considered to be phosphorylated. Here's two visuals. Up here, I took ATP, I broke off a phosphate, remember ATP to ADP plus PI? So look, this phosphate ends up binding to this membrane protein, this carrier protein. This protein has been phosphorylated, okay? It's going to change the shape. This carrier protein is going to snap from being open to the intracellular to snap to being open to the extracellular. And we're going to transport that solute. That's the work that just happened. It happened because we phosphorylated the protein. Once we snap open, now that inorganic phosphate can come off and it's free to rebind to ADP and we'll have ATP again. The bottom track over here, if I follow this arrow, okay, this is going to be mechanical work. So I've got a vesicle, it's going to bind to a motor protein, and I'm going to use ATP to phosphorylate that motor protein to advance it along the cytoskeleton. I'm going to use it to advance it along and move it. And that's going to cost me, I'm going to phosphorylate it. Once it moves, the phosphate falls off, I'll have ADP and PI, they can couple back together, and I'll have more ATP. Okay? Just two examples. I wanted you to see them in case you were struggling to find them. Beautiful part about ATP, it's renewable. So ATP is totally renewable in that we can regenerate it by phosphorylating the ADP. So the exact thing I did through hydrolysis, where I pulled the phosphate off of ATP, now I can add it back on to ADP, and I'm right back at ATP. Okay, so the energy to phosphorylate ADP comes from other catabolic reactions in the cell. The potential energy that temporarily gets stored in ATP is what's going to drive that work. Here's your visual. And here's a question. Counting to 5, you work it out. I will say that on the first one, I prefer hydrolysis to exergonic. I'll give you credit for exergonic, so when I forget I said that, somebody remind me. But I really like hydrolysis better because that reaction just doesn't happen without the presence of water, okay? And then your second one stands pretty well. Another one. And the answer? Remember, phosphorylation is just that addition of the phosphate molecule or the phosphate to a molecule to a reactant. So I'm building that phosphate back on, and when I add it, I add another bond, and I increase that potential energy. Okay. Catalyst. I introduced catalyst to you in the the macromolecules chapter. And a catalyst was any chemical agent that sped up a reaction. And we specifically talked about enzymes that chapter. I remember talking about things like lactase and sucrase and catalase. So an enzyme is a protein. It's a protein that causes catalysis or is a catalytic protein. Remember that all enzymes are proteins, not all proteins are enzymes. So what ends up happening in the case of sucrose is the hydrolysis of sucrose by the enzyme of sucrase. So I have to have sucrase present to speed it up, will be an enzyme catalyzed reaction. The same thing happens with lactase and lactose, same sort of thing. But here's the visual example for sucrose. So sucrose is the sugar in the presence of water and an enzyme called sucrase that will split into glucose and fructose. The presence of sucrase allows that and that reaction to go more quickly. Okay. Will this happen without the presence of sucrase? Yes, it will. Just really, really slowly. In comparison, really slowly. All right. So I'm going to pause here. We're going to come back in at activation energy barrier, but this will be part 1 of the enzymes and energy chapter.